My name is Minalini, and I'm a research scientist at the San Diego Zoo Wildlife Alliance. Thank you for the opportunity to speak to you today. The 36 recognized global biodiversity hotspots, which are located both north and south of the equator, occupy just 2.4% of the Earth's land surface, but they are home to over 50% of endemic plant species and 42% of terrestrial vertebrates. Yet, 50% of the world's 187 genomic initiatives are based in the United States alone, and 19% in Europe, regions with comparatively lower biodiversity than those in the global south. And meanwhile, many of the world's most biodiverse regions lack the genomic infrastructure needed to support conservation efforts. So, this mismatch is not really just about technology. It reflects persistent obstacles in funding, access to training, and access to infrastructure that conservation programs face in traditionally ignored markets. But the future is bright. With innovations in portable, affordable sequencing tools, we now have the opportunity to democratize conservation genomics that previously relied on exporting samples to the north for sequencing. So in this talk, I hope to share our efforts to build and test a fully field-deployable nanopore-based pipeline designed to be low-cost, modular, and accessible so that others can adapt, and scale it to protect biodiversity where it's needed the most. On the surface, when you consider nanopore technology, we're talking about shrinking the size of sequencing tools. But in reality, we're changing where and how genomics happens. These devices let us bring science to the source, cutting out delays and empowering local teams to act on data in real time. It's a fundamental shift from centralized control to distributed, field-ready capability. This opens the door for models like the In Situ Lab Initiative, which are embedding genomic capacity directly in conservation landscapes by shortening the learning curve involved. The In Situ Lab Initiative, or the ISL, is a global network of field-based labs using portable genomic tools to support local conservation and capacity building. Each hub is operated by in-country researchers embedded in biodiverse regions with active and different conservation needs. So the ISL was born out of a simple idea. Biodiversity hotspots need the same access to genomic tools as any high-end institution, without depending on export permits or expensive centralized facilities. These labs are embedded where conservation decisions actually happen and where samples can be collected. Each site is operated by trained teams of scientists using nanopore devices to generate real-time genomic data quickly. What makes this model different is not just the portability of the tools, but that it stays rooted in the countries where the biodiversity exists. At the end of the day, it's really not just about generating data, right? It's about answering questions that conservationists and enforcement agencies are actually asking. So whether we're working on bears or birds, bats or frogs, the core needs are similar: identity, origin, sex, kinship, and population structure. These answers guide everything from translocation and breeding to detecting illegal trade or planning reserves. The power of our pipeline is that it can flexibly address all of these questions across diverse taxa. We developed two parallel paths for building marker sets to achieve these goals, depending on which resources we have at hand. If we have access to many high-quality samples across a species' range, we can build a genomic atlas and extract SNPs based on population-level variation. We use a streamlined pipeline called MPCR Select, which applies basic QC steps like thinning and missingness filtering to identify around 500 high-performing SNPs, and it's the most efficient and easy-to-use pipeline to date. These can be optimized to minimize nucleotide diversity or maximize population differentiation, depending on whether we're targeting individual identity or species, subspecies, or population splits. We always validate the marker set using PCA or clustering to check how well they separate individuals or groups. On the other hand, if we only have a single good sample, we go straight to building a reference genome using the P2 solo in country. Then we use a simple and fast pipeline to find STRs using perf, filtering for repeating lengths between 11 to 20 base pairs and focusing on foamers with high base complexity, a process that was developed and fine-tuned by the FOGS project. From these, we identify SNPsters, which are short tandem repeats linked to informative SNPs. These give us strong discrimination power and can be used to build pedigrees for conservation programs that need it. So we're working towards making both paths completely nanopore compatible and validating them against gold standard sequencing pipelines Ultimately, both of these will get us to low-cost amplicon panels that can be used for fast field genotyping. So we use both SNPs and SNPsters depending on the question at hand. SNPs are fast, cost-effective, and great for species ID, sexing, and individual assignment, even from low-quality samples. But they're limited when it comes to relatedness and pedigrees, especially when we focus on using small panels so that we can have just a single test. And sexing markers are not always guaranteed because many existing genomes for non-model organisms don't have an assembled Y chromosome. SNPsters, on the other hand, are more powerful for kinship analysis. They combine STRs with nearby SNPs, but they require a bit more complex bioinformatics work to generate genotype data. And by definition, since they deal with kinship, they need to be applied across a population to produce the right outputs. So our workflow starts with species ID, either built into the SNP panel or through a rapid barcoding run using a protocols.io pipeline that you can see on the right, and something like ng species ID to generate consensus sequences. Once we've assigned samples to individuals using a quick SNP-based test, we can then run SNPsters on one sample from each animal to dig in deeper into the population genetics of the study system. Marker validation is a multi-step process, exacting and time-consuming, and it's really critical to how much a pipeline is trusted and how easily it will be adopted by a new user. So we take it really seriously and we give it time and attention. So first, we run PTEXP1, which is a primer testing experiment that uses 2 individuals, a male and a female. We test 5 concentrations of a primer pool across 6 to 10 annealing temperatures. We deliberately avoid using ramped PCRs so that this can be done using pretty basic thermocyclers. This increases accessibility, though ramping is totally feasible if the equipment is available. Next, we run SNEXP1, which is a sensitivity test. We use 3 to 4 individuals across multiple tissue types, blood, scat, or hair, depending on the species, and we run 10 different DNA input concentrations per sample. So this helps us set a minimum DNA threshold per sample. For example, feces often contain less than 1% endogenous DNA, so they tend to require more starting material than a blood sample. Once we've identified the optimal PCR conditions and the minimum input of DNA that's required, we run PTEXP2. This uses a larger sample set, so 10 to 20 individuals, including those used in the original marker discovery, as well as some new, related, and unrelated individuals from different populations. This tells us which markers consistently amplify their heterozygosity and overall genotyping success. We iterate this sometimes up to 4 different marker pools per species. We tweak primer concentrations, we even remove markers, but anytime we make any change to the pool, we have to restart all our experiments and revalidate the pool. Finally, the finalized marker set is tested against potentially confounding or co-occurring species to check for specificity. So in the case of the Andean bear here, the purple taxa are animals that occur in the same region. And if we're collecting scat from the wild, we might accidentally collect scat or hair from these animals. The blue animals are closely related species, so other bears, and those in orange are the ones that our lab is working on currently, and we want to make sure that none of our markers are going to amplify against any of these. So every validation and genotyping step in our pipeline, both in the lab in San Diego and in satellite labs at partnering sites, is performed using Nanopore. Not only is it affordable when paired with our multiplexing strategy, but it affords incredibly fast turnaround, so we can push forward every week quickly without a supporting genomic score. The figure here walks you through our full workflow from DNA extraction to bioinformatics, and our indexing strategy, which is highlighted in step 3, is central to keeping this efficient and cost-effective. If you're interested in replicating this approach, we've made the full protocol available on protocols.io, which you can access via the QR code to the right. To wrap up, I want to share a few of the protocol tweaks that have made this pipeline work reliably across species and sample types on a Nanopore platform. We optimized everything using single temperature PCRs, avoiding ramped protocols, so that these could be run in basic field labs where overly complex or modern PCR machines may not be available. That said, if you have touchdown-capable thermocyclers, this can really help improve genotyping performance. We increased our library prep input to 400 fmols per barcode and doubled the bead cleanup ratios for fragment sizes under 350 base pairs. This helps to reduce primer dimers and can clean up the background before loading. And keeping things under 350 base pairs makes them more likely to work on hair and scat. Ligation times were extended to 20 minutes to compensate for the short fragment size, and loading concentrations were adjusted for flongal and minion flow cells to 50 or 100 to 200 f-moles respectively. We also noticed better results after adjusting flow cell rest times. So 10 minutes for flongal flow cells and 30 minutes for minion flow cells, which allowed molecules to find the tethers more effectively. On the analysis side, modifying mapping parameters made a big difference. Because Q scores are generally lower for shorter reads, we avoid strict matching and relax some parameters to avoid unnecessary read loss. You can see that in the quality by length plots, short reads can still give pretty high quality if handled correctly. The nanopore compatible pipelines that we've made will go public soon when we kind of hit a satisfactory level of perfection. It's really hard to know when to stop trying to perfect the pipelines. So this is work that has been made possible by so many different team members, contributing their efforts to finalizing and perfecting different aspects of the pipeline. Our partners in the field inspire us every single day to push on through just one more validation experiment to make things as easy and reliable in the field as we can. Finally, we're grateful to the ONT team here in California who go above and beyond to support our research group, even though we are far from being their biggest customers. So thank you for continuing to support small groups like ours.